to track.
Bless you.
And we were talking about the liver. Hadn't we gotten to talking about the liver stored a lot of valuable things? Okay. But on the flip side, the liver is also an organ of detoxification. So one of the things it's constantly removing is when you break down protein. Proteins are made of an amino acid. And the subunit amino acid has that name because every one has in it at least one amino group. Well, here is an example of an amino group. And if that floats around in your bloodstream, the amino group most commonly takes up the form of NH3. It can be in there as NH2 or NH4, but most commonly it's that. That is called an ammonia. And we mainly get them from degrading proteins in our body. If you built up high concentrations of ammonia in your blood, that would make you become basic, and that would be toxic to tissues. So that is combined with that. So there's an amino group, and there's another amino group, NH2. Each of those have both lost a hydrogen, so there's two of the hydrogens. There's your carbon dioxide, it's lost an oxygen, so there's that. So this ends up becoming urea and water, and the next place that's going, and some will be eliminated, is to your kidney. So every time you go through your kidney, 1/2 of the urea that enters your kidney, 1/2 of this molecule, will be dumped out into your urine by your kidney. So if at that moment in time you have a lot of urea in your system, then a lot will be dumped out in your kidney. If at that point in time you have a little bit of urea in your bloodstream, then only 1/2 of that smaller amount will be dumped out in your kidney. So it's always a 50% equilibrium between urea that remains in your body and urea that goes out in your urine.
Ms. Halls, what does that say after the two, the H2?
That's just a
Okay.
nothing. I think we talked about that last time. So it has many valuable things stored in the liver, but it has some toxic things made in the liver, and so some people go, liver is an unhealthy organ to be ingesting as a food, but I don't know. It's one of those things you have to make up your own mind about. Many proteins are made by your liver that are important to us. These first two you had in your first test, fibrinogen and prothrombin, are examples of blood clotting proteins. So if you need to clot your blood, you can. But notice the liver also takes care of the other side of the story. It also makes a blood thinner, an anticoagulant, heparin. Those are the largest sized proteins that are in your bloodstream, the clotting proteins, but the largest amount of protein in your bloodstream is the albumin that gives you viscosity, makes your blood resist being real runny. And we're going to find out that albumins are carriers for hormones also in a future topic that we come up on. The liver was a soft, mushy organ, so as a fetus, before you were born, it made blood cells, and still it's got a lot of sinusoid spaces in it that store a lot of blood, so it also has to do with blood volume in an adult. You're going to see today in lab that the sinusoid spaces in the liver periodically, not continuously, but periodically have a little flat cell plastered to the edge of the sinusoid where the blood is, and those cells that look kind of squamous are called Kupffer cells, and they phagocytose things that are in the liver. So the liver is a filtering organ, and the Kupffer cells help to modify the blood as it passes through the liver to make it healthier as it is recycled back out to your body. Kupffer cells are a member of a lot of phagocytic protective cells. So they're all collectively in the name of cells called reticuloendothelial cells. For instance, last semester, when you studied the reticular connective tissue in a lymph node, there were a lot of phagocytic cells in there that were called reticulocytes. So this is just another member of that phagocytic group that is cleaning body fluids as they're filtered through these soft, mushy organs. So it is specifically known as a Kupffer cell. The liver stores cholesterol, and we've already talked about that before, so these are just notes from last time when I was clarifying which cholesterols are good and bad for you, so that's not for today. We've already gone over that. Now, when you go in lab today, you'll be looking at a slide of liver, and liver is made of repeated subunits, and each subunit is called a lobule, but you kind of have to stay on a low overview power to even be able to kind of pick out these little blobs or lobules of liver tissue. At the center of each one is a drainage vein whose name is a central vein. So that's one central vein. And you see another one right there. So there would be literally hundreds and hundreds of these in a liver, but what happens is blood is brought to the liver. And if you recall, blood is brought to the liver as clean, oxygenated blood and deoxygenated, dirty blood that's coming from your visceral organs that needs to be detoxified. So these sinusoid spaces, these gaps between the cells, are a mixture of hepatic artery blood that is clean and oxygenated getting dumped together with hepatic portal vein blood that is already deoxygenated and dirty. That's why we don't call these typical capillaries, because they are mixed blood coming in. They're called sinusoids. And so where does the blood come into that hepatic artery and hepatic portal vein? At the edges of each lobule. So here comes the dirty blood in the hepatic portal vein, and there's the little muscly wall of the hepatic artery. That's the clean blood. Then it dumps and percolates towards the center. So I didn't draw it each time, but every one of these lobules will have branches like this of those three vessels. They're just branching smaller and smaller and smaller as you come down to a lobule. So each one around its periphery will have a set of those. This group or triplet of hepatic portal, hepatic artery, and bile duct is collectively called a portal triad, because three things are traveling along together. So that's how blood gets in. Blood gets out by going out this central vein, and at the top of the liver, all the central veins converge to form the hepatic veins, which dump into your inferior vein cava and take you back to the heart. So last time we were in here, we said the majority of cells are cuboidal, and those are the hepatic cells, and those are the cells that made bile. These are your bile-making cells. The Kupffer cells are the really dark-staining flat cells that filter as blood percolates through the sinusoids. Are you okay on the liver construction? You would just have more and more of these. Okay, now we're going to go down to the gallbladder then. By the way, here is a liver, and what we've done is the gallbladder would have been embedded up underneath the lobe of the liver. We just peeled it loose and dropped it down so that you can see the association of the various ducts. So GB, gallbladder, its role, it doesn't make anything. It concentrates the bile that the liver made in its hepatic cells, and it stores it so that there is a big load of bile in a gallbladder. And therefore, if that's its goal, it has to be able to store a lot of this So it has rugae, gathers, that it can unfold. So it really is adapted for storage. We also think, just like we thought the liver was an outgrowth of your small intestine by way of epithelium, we think the gallbladder is also an outgrowth of epithelium for the duodenum, and we think both of these glands kept their ducts all the way over to the small intestine, the duodenum. So it depends on what you're doing at the moment. If at the moment you haven't eaten any fatty food, then the liver is just using its hepatic cells to make bile, and the bile needs to get over here to the gallbladder, so we need to store it. So the storage route would be, you take the little tiny bile ducts, so sometimes they're called biliary ductules, ULE ending, but even if you call them bile ducts, and they're going to converge and make the major duct from the left side and the right side, and so those are known as your left and right hepatic ducts, L/R hepatic ducts. And where those converge, the left and the right, they become the common hepatic duct. And now you turn the corner and you got to go to the one duct of the gallbladder, which is called the cystic duct. And so then the bile is stored in the gallbladder. Cysto means bladder, so that's telling you gallbladder. That's its duct, cystic duct. Now, if I ate fatty food, the fatty food would trigger the gallbladder to contract, which would take that very concentrated, massive volume of bile and shoot it into the duodenum where the fatty food has arrived. So that would be the secretory route. So it'd be gallbladder to the cystic duct to the duct that dumps into the duodenum, which is your common bile duct. So if you've had your gallbladder removed, a lot of times what they will do for you is take the hepatic duct or common hepatic duct and make sure that that is still maintaining a connection over to your duodenum, so that you get a little bit of droplets of bile that your liver is making that are dripping into your small intestine. The only problem with that is you're not going to have this big load of stored up bile there. So what does bile do for you? Bile, and you do an experiment with this in lab today, acts as an emulsifier. Pretend you have a stick of butter, and if you cut up the stick of butter, you could touch more surfaces of the butter all at one time, more surface area. That is what emulsification is like. Fatty foods are gunky. They tend to stick one fat molecule to another fat molecule. So we spray them with bile, and the fat molecules tend to let go of each other, and then enzymes that are in your small intestine to actually digest the fat can now get to them and do a better job. If you have your gallbladder removed, a lot of times you have what is known as a fatty stool. A fatty stool is you're not digesting the fat in your digestive tract. A fatty stool translates into diarrhea. And so a lot of times when people first have their gallbladder removed, you'll be on a really strict diet. They will say, stay away from red meat, beef and pork. Eat very white, like breast of turkey, or a really white fish, not a real oily fish like salmon or some of those that are supposed to be really good for your health because they're full of a lot of oils and greases. So then you eat your white chicken breast or turkey breast or your white fish, and you see how you get along with that. And if your body is behaving okay and you're not having to run lickety-split after every single meal that you have, then they might say, well then, try a few other things. And so you might go to some dark meat of the chicken, or you might go to pork tenderloin, which is a really white, not real oily pork. If you get along with that, they may say, well then try one of your foods that you love or that you haven't been able to have, like a hamburger or something, and then see how you do. Some people do very well, and their body will kind of gradually get over the fact that you don't have the bile, and they can eat comfortably and enjoy their meal without incident or accident. But you could have two people in the same family, a husband and a wife, like grandma and grandpa, both of them have their gallbladder removed, and he can eat anything he wants and she can't. It just, there's differences in body chemistry, and some people are very sensitive to the diuretic condition after they've had their gallbladder removed. Other people, it's when you eat something that you haven't had often, like a spaghetti or something with a lot of spice in it, or something really fried food, or something very oily, something like that, then sometimes they have trouble with it. But just have to try it and see how you get along with it. All right. Any question on gallbladder? It's a rather simplistic organ. Then we're going to come down to pancreas. Now, I don't have pancreas drawn in this picture, but your stomach would have been right over here, and your pancreas is sort of between the stomach and the small intestine, and it kind of strings out like an oak leaf out this direction. The long and short of it is, if I drop the pancreas down, and I had a pancreas in the picture, like a big hamburger meat oak leaf, it also has a duct, and its duct comes out of the pancreas and at the last moment unites to the common bile duct as well. So that point at which the common bile duct is being united with the major pancreatic duct of your Yersun, there ends up being an enlargement in the tube at that point. And then a nipple sticks out into the small intestine. So the enlargement is called the ampulla of Vater, the big space. The big space is the ampulla of Vater, and the nipple is the papilla of Vater. And encircling this is a sphincter muscle that if it is stimulated, it will shut the tube so you can't have secretions entering the duodenum. So the muscular control is called sphincter of Oddi. Sphincter of Oddi. And it would be in a position right about here, where it could close off the tube. But you had done a diagram of this previously in the lab, but it seems to be a constant source of confusion. What's the ampulla? That's the big space. What's the papilla? That's the nipple that sticks into the duodenum tube. And the muscle is embedded in the wall and would encircle the whole thing. That's the sphincter of Oddi. Well, anyway, if you look at the pancreas is what we call a mixed gland, because it has some parts that have ducts, exocrine, over to your small intestine. That's secreting digestive secretions. That says exocrine part. The other part is an endocrine part. And from Anatomy 1, an endocrine, INE, gland is a gland that doesn't have ducts. It is ductless. Ductless. So how does a ductless gland deliver its secretion? They diffuse out into your tissue spaces and eventually hit a blood capillary, and so it's your bloodstream that's going to deliver the hormones of the pancreas. And so we'll spend a lot more time on the hormone part of this story later when we're in our hormone unit. So in lab, you studied this, and so all of the background cuboidal cells were pancreatic acini, and they're the ones that make pancreatic juices, which are digestive enzymes. So they're the exocrine part, the part with ducts. So they're carbohydrases that work on sugars and starches, lipases that work on fats, proteases that work on proteins, nucleases that work on DNA and RNA, and a buffer, bicarbonate ion, because guess where you are? You are in the first part of the small intestine. You are receiving all that highly, highly acidic chyme from the stomach. So we're trying to bring it back up closer to neutral by buffering it with the bicarbonate ion. So these are the things we're interested in now, and we'll come back and talk about specific examples of these digestive chemicals in a minute. Do you have any comments about the digestive part, though? The other cuboidal cells, so if all the background on the paper were the pancreatic acini, then every now and then clustered in there is a group of a dozen or so cuboidal cells that are insulated and surrounded by the darker pancreatic acini. These little isolated clusters are islets of Langerhans, and they produce various glucose controlling hormones. And the glucose controlling hormones are listed over here. In this little cluster, there are 3 different kinds of cells. Some cells are called alpha cells, and they make glucagon. And glucagon makes you raise, elevate blood glucose. In other words, it keeps sugar in your bloodstream. How come? Because of your brain. The only food a normal brain will use is glucose. And if you don't give it its glucose, you're going into a coma. So that's a second reason that you go into a coma. Getting too acidic will put you in a coma because that inhibits your nervous tissue, and not having any glucose will put you in a coma. The competing cells in here are called beta cells. And by the way, alpha and beta are just like calling it cell A and cell B. Alpha and beta are the first 2 letters of the Greek alphabet. So it's very simplistic. So beta cells make a competing hormone called insulin. Insulin gives the sugar to your body cells. Therefore, it's lowering the sugar in your bloodstream. So this lowers blood glucose, so that your body cells aren't having a starving, craving, appetite fit. Okay? Delta cells make a regulator, and it is called somatostatin. Put an N on it. Somatostatin. Statins, you know, to balance things. This statin is supposed to balance your glucagon and insulin levels so that you're doing this properly in your body. They don't know a whole lot more about somatostatin, but that's its job to kind of keep these in balance. So we will be talking more about the complications with glucagon and insulin in the future, but that's what they're for. And by the way, somatostatin is produced by a third cell called a delta cell. Are we all right? Okay. I think we have now gone over the structures of the digestive organs and the accessory glands. Now we want to talk about the enzymes and where they come from. And let me tell you, the copy machine wrinkled a bunch of these pieces of paper. So if it looks like you've got one thing twice, it's because you've got a bad copy, and some of it didn't print because of it wrinkling the long paper. So we've already done this earlier in the term under the bloodstream, so this is just a review right here of something you've already had, so we'll talk about that in a minute. Are you coming to the afternoon one? I know, I know. Now, the rest of you are getting one that the back may be crappy, but the front's good. And I've already given you a good back, so you've got front and back. You're done. Subtract. Okay, so what we want to start on is the handwritten one I gave you first. Because what we're trying to decide is what are you going to have to digest in your digestive tract. So you ingest carbohydrates. There are only two kinds of carbohydrates the human ingests. They are sugar and starch. And the most common starch of a human is called glycogen. Glycogen starch. And starch is made of nothing but glucoses bonded together. We learned that in your lab last week. So you're going to have to break this glycogen starch down to individual monosaccharides. In other words, it's got to be digested down to the monosaccharide, which is sugar, which is also known as dextrose. Two names for the same sugar. In the process of going from the starch that is made of hundreds of glucoses bonded together, there will be some times that several of those glucoses will still be stuck to each other. That forms a variety of sugar pieces called reducing sugars. So examples of reducing sugars are dextrins, which is 3 or more glucoses hooked together, maltose, which is just 2 glucoses hooked together, or glucose itself, which is a single sugar. That's what you tested for with Benedict's last week, is the presence of reducing sugars, which was evidence that you were in the process of digesting down a starch. Is everybody okay on that? The deal is this: you cannot absorb a carbohydrate until you get it down to its monomer, its individual monosaccharide, single or simple sugar. So that's what we're trying to get down to. Okay, so that's one food. Or you could have ingested sugars outright from your Snicker bar or whatever you ate. So you could have ingested glucose itself, in which case you could absorb that right now. You could ingest lactose, which is a disaccharide. It's 2 sugars bonded, and it is called milk sugar. That's where you find it. And so it's in dairy products. So you hear about people that are lactose intolerant. They may not have the enzyme to break down the lactose, and therefore it may give them projectile vomiting in a newborn baby or something like that. Then what would they do? They either have to supplement the milk and give them enzymes to help them digest it, which sometimes as you get older they'll do, or you take a different kind of milk in your diet, like goat's milk or something that you do have the enzymes to break down, and it won't give you that kind of GI tract distress. But let's say you don't have that problem and you can use milk sugar in your body, then that disaccharide, when broken apart, becomes 2 different monosaccharides, galactose and sucrose. We'll say something about all these monosaccharides in just a minute. Or you could have ingested a disaccharide, which is a grain sugar. That's called maltose. Maltose, when you break the 2 sugars apart, is going to become 2 glucoses, because that's what maltose is, 2 glucoses bonded together. Or you could eat table sugar. That's what's in your Snicker bar. And when you break that disaccharide apart, one is a fruit sugar fructose, that's a monosaccharide, and the other is the monosaccharide glucose. So these are the end products over here on the far right. It happens that every one of these monosaccharides that we absorb is C6H12O6. All of these monosaccharides of human absorption. And those are different from each other only because of how the hydrogens and oxygens are arranged within the molecular structure. That's what makes them different. Those are all called hexose sugars from Anatomy 1. If you have 6 carbons, you're a hexose. Okay, so we know what we're trying to get our sugars and starches down to, individual monosaccharides. Is that clear? Okay, lipids, fats. The neutral, most common fat of our diet is called triglyceride. And when it is digested down, and you're doing an experiment on this today in lab, it ends up being 3 long fatty acids and a small little glycerol. These fatty acids I grossly oversimplify in class. I just make them look like they're 6 or 12 carbons long. You call these hydrocarbons in Anatomy 1 because they had a heck of a lot of hydrogen stuck to carbon and just a couple of oxygens in it. These fatty acids could be hundreds of carbons long, and they typically are. So there is no way something that big is going to go through the tiny little pores between the cells of a capillary. So fatty acids and glycerol are most often absorbed in that structure that was in a villus that had a big open pore at the top. That was your lymphatic lacteal. And they go in there, and so much fatty food is absorbed in the lacteal like that it is a whitish liquid called chyle. Remember that story? C-H-Y-L-E, chyle. That's the fatty food entering your lymphatic system. This is a villus in your small intestine. But those are considered the smallest subunits of fat. Now, if you're a discriminative reader, you will hear about something called a glyceride that you can absorb into your digestive tract. That is, or from your digestive tract into your circulatory system. That is a glycerol with one fatty acid still attached. But it's usually a real short little fatty acid. But that is the exception to the rule. The bulk of your lipids have to be broken down this far for you to get them into your circulatory system. So I'm not going to deal with the exception, but I'm just saying, for those of you who are doing the reading, you're correct. There are a few little tiny fatty acids that get to stay on the glycerol. If you eat protein, that has to be broken down to a single amino acid, a single subunit. So there's a theme here. We're getting down to the individual monomer, the individual building block of each of these different organic groups. The last organic group is your genetic material, nucleic acids, and there's DNA and RNA types of nucleic acids. They have to be down to their single monomer, which is a nucleotide. And lucky for us, anything you eat or anything known in the world, universe, or wherever, the only kinds of nucleic acids we know are nucleotides with the same bases that we have: adenine, guanine, cytosine, thymine, and uracil. So we know of no others. So it doesn't matter whether you ate lettuce or cow hamburger or whatever, when you break its DNA and RNA of its cells down, it's just going to be nucleotides with the same bases that you need to build your own genetic material. Now those are the only 4 groups you have to digest, the organic groups. But there are 3 other things you die without. So you have to soak these things up from your digestive system even though you did not have to digest them. Minerals, vitamins, and water are essential, that they just don't require any digestion. Now, the mineral or the vitamin might be buried down in the depths of food, so you might have to digest these other 4 things off of it in order to release the vitamin or mineral. That's why even when you're down in your colon, your large intestine, you may be still soaking up the last residual vitamins and minerals, because the bacterial action on waste and food might continue to release a few last particles that are useful to you. Are we okay on that? All right, so these are the things we want to uptake. The individual monomers down the right and the 3 vitamin minerals and water at the bottom. Okay. So on the other side of this sheet, what we have done is written your digestive tract across the top. Mouth, esophagus, leading to your stomach, pyloric sphincter, leading into your small intestine. I gave 2 columns for small intestine, mainly because I ran out of room in 1 column, but everything starts in the duodenum. And whatever starts in the duodenum is probably continuing on in the rest of the small intestine. And then you go through the ileocecal valvar juncture, and you're in the large intestine or colon. And once you're there, there's no more digestion. There's just possible absorption. So we'll come back to that. So then down the margin, I'm first going to take you through that whole digestive tract and see what happened to the carbohydrates. Then take you through the whole digestive tract, what happened to the lipids, what happened to the proteins, what happened to your DNA and RNA nucleic acids, and then I have some extra little comments to make as well. So you see how the sheet is set up? This is one of those kind of things. This will give you the information. Then you have to decide how you want to organize it. Do you want to make flashcards of how far you've digested the food and what's the enzyme that's going to take you to the next step? So you need to know, what food did I begin with? What's the enzyme? What did it leave me with? If that's not down to an individual subunit, now you need to know, okay, now what's that piece called? What's the enzyme I need to break it down further until you finally get it to one of those subunits we just discussed on the other side of the page. Are you with me? Okay, so let's try this out. We're in your mouth. There is only one ding-dang enzyme in your mouth. What is it? Salivary amylase, also known as ptyalin. So it only works on what food group? Starch. So that's why it says no action on a monosaccharide or a disaccharide if it's in your mouth, because there is no sugar splitting enzyme in your mouth. There's a starch splitting enzyme. So starch is the only thing worked on. It's worked on by salivary amylase. Now, the name ptyalin only applies for salivary amylase, not for amylase made in other organs. So we'll get amylase made in your small intestine and amylase made in your pancreas, but the one called ptyalin is only made by your salivary glands. So, and notice the P is silent and the Y is long. So it says it came from your salivary glands, and it breaks starch down into maltose and dextrins. But look, very little of it. Of the 100% starch you eat, only 5% of it is broken down at this point. So it's barely worth mentioning, but it's true. Then all that food went down your esophagus. So anything that started in your mouth is continuing to digest in your esophagus, and it plops into your stomach, which is highly acidic. But if your stomach is full of food, digestion of starch may still continue for a little while, because you have to have those 15-second tonic mixing waves to mix your starchy food that's just arrived up with the acids of your stomach before it will denature the salivary amylase. So you might get 15 minutes or so of starch digestion, but it still won't account for more than 5%. But very quickly in your stomach, you're going to denature that ptyalin or salivary amylase, and there won't be any new digestive action on starch in your stomach. So not much on starch. Now the starchy food and sugars move down to your small intestine. The duodenum is an important site because it has all those ducts coming in from your pancreas. Your pancreas makes all kinds of digestive enzymes. So pancreatic amylase is sprayed in on your starchy food. So we're starting starch digestion all over again. Pancreatic amylase has an asterisk because I'm making you a list on this chart of everything that came from your pancreas. So every time there's another thing from your pancreas, there'll be another asterisk on your sheet. So pancreatic amylase is called amylopsin, A-M-Y-L-O-P-S-I-N, amylopsin. And again, it will start digesting the starch down. And so if it gets it broken down to a disaccharide, it's maltose. Now, there is also a little bit of amylase that comes from intestinal cells. And I told you this before, the way they think the intestinal cells, which are simple columnar epithelium, release their digestive enzymes is they think they exfoliate. They let go of their basement membrane, and as they're coming up the crypt of Lieberkühn gland, they decompose. And in the process of decomposing, that's when their enzymes are spilled out into the small intestine. So that's why it's talking about decomposing intestinal cells. They've already exfoliated or let go of the basement membrane. So we have amylase from 3 places then in your body. From your salivary glands, from your pancreas, and now from the intestinal cells in the crypts of Lieberkühn. Are we okay on that? All right. We may have only gotten your starch broken down to disaccharide. So, or you may have ingested some disaccharides and they've been traveling mouth, stomach, mouth, esophagus, stomach, and now they're in the small intestine. Those disaccharides are going to get hit by enzymes made from your intestinal glands. Again, the name of the intestinal glands are crypts of Lieberkühn. So if the name of the enzyme is sucrase, guess what disaccharide it's going to digest? Sucrose will be broken down by sucrase. And when it's broken down, it becomes glucose and fructose. So this is the end monosaccharides it becomes. By the way, all these have a glucose in it. So this one's glucose and fructose. Now they're absorbable. If the name of the enzyme from your crypts of Lieberkühn glands is lactase, guess what that acts on? Lactose. And that's the one that was made of something that sounds like lactose, galactose and glucose. Those are 2 monosaccharides ready to be absorbed. If the enzyme is maltase, then it's working on maltose, and that's the one that's made of just 2 glucoses. Now they're ready to be absorbed. Do we want any of the sugar to get away from us? No, we want 100% of this good nutrient to go into your bloodstream. So we're going to use energy, ATP, to force all of the monosaccharides out of the small intestine and into your bloodstream. That's active transport because we're pushing it all in one direction. So there's no sugar left by the time you're in your large intestine or colon under normal circumstances. Any confusion there? Yes?
In that column, it's just showing us how each sugar is like digested?
Yes, each disaccharide sugar that you could have eaten would be digested by enzymes from your crypts of Lieberkühn glands. Yes, that's exactly what it's showing you. What? You look like...
Is it, they're just this disaccharide? It's not talking about...
If it were a dextrin, a string of sugars, then your amylases are going to work on that until it gets it down to maltose, a disaccharide. Then to get from a disaccharide down to a monosaccharide, you need one of these 3 enzymes from your crypts of Lieberkühn. Okay? Is that clear, everybody? So we don't typically eat anything other than a starch, which becomes maltose, and now, or disaccharides. And we have enzymes to break down our disaccharides. All right? So carbohydrate digestion is pretty simple. You need to know the amylases and 3 disaccharide splitting enzymes. But you need to know who secreted them, what gland, and what the food becomes. All right, let's look at fats. Fat digestion's not complicated. Fat absorption is your problem because it's gunky. So we'll come back to that in a minute. The most commonly ingested fat is triglyceride, and in the American diet, because we can afford pork and red meat, we're considered rich diet. We have money to buy that kind of good food. We're not just eating roots or soy bean as our protein. So we tend to have too much triglyceride in our system. But nothing in the mouth will act on fatty foods. And it does say in your reading that the stomach, gastro means stomach, makes lipase, so it's called gastric lipase. But the minute the acid condition of the stomach hits the gastric lipase that the gastric pit glands have secreted, it's denatured. So is there any appreciable amount of lipid digestion in your stomach? No. So it's questionable why we even make gastric lipase if it's going to get denatured so quickly. So nothing there. So we're down to the small intestine. These are both small intestine columns. So the first thing is when that fatty food is detected, your gallbladder is going to contract. And your gallbladder then sprayed bile onto the fatty food. So that now, like me taking the chunk of butter and cutting it up with a knife, you're going to separate those fat molecules. That's emulsifying the fat. Also, the pancreas secretes lipase. So there's an asterisk. There's an asterisk because this is another thing from your pancreas. Pancreatic lipase is called steapsin. That's what you're using today in your experiment is pancreatic lipase. And it'll break those triglycerides down to fatty acids, FA, and glycerol, GLY, glycerol. You also have intestinal lipase. So that would be from your crypts, Leberkuhn again. So it'll act in the same way as the pancreatic lipase. And so they're ultimately all broken down to fatty acid and glycerol. And there's that one exception. If a fatty acid stayed hooked to the glycerol, it's glyceride, but that's unusual. And if you ate food that had vitamins A, D, E, and K in them, those are fat-soluble vitamins because A, E, and K are lipids, and D is a steroid, a cholesterol compound, and that's still gunky, fat-soluble. So however I tell you we are going to soak up your fatty acid and glycerol, that is the same method you'll use to absorb the A, D, E, and K because they have the same problem of being gunky. Okay, so what I want you to do is in the other packet that you have, flip the next page because it tells you how we absorb fatty food. Just flip that over. Everybody got it? So it's just reminding you, we've gotten food broken down this far. Vitamin A, D, E, and K just had to be released from whatever it was tangled up in. And then we have fatty acids, and we have glycerols, and maybe an occasional glyceride. These are going to have to get into your columnar cell, your simple columnar epithelial cell. So that's what I'm representing by that one tall cell. I'm being very lazy. I did not draw you a lot of them, but the whole villus is covered with simple columnar epithelial cells. So in order to get these end products of fat digestion absorbed, they first have to be sprayed with bile. So this is another use of bile. Remember, before you even digested the fat, you had to spray it with bile to get one fat not to stick to the fat next door. That was emulsification. We've already talked about that. Now we're digested. We're way past that. But these end products have to be coated with bile to become water soluble, because they've got to be water soluble in order to get into the columnar cell, because they move along with a lot of the water that you're supposed to be eating in your diet, 8 glasses of water a day. So they're now renamed. These end products of digestion that are bile coated and water soluble are called micelles, and that's what enters your columnar epithelium. So know your vocabulary words. Now this is going to kill you. The minute we get them across the cell membrane of the columnar cell, so they're inside this columnar cell, the ding-dang 3 fatty acids stick back to the glycerol and reform your triglyceride. The only reason we did all that digestion with lipase was to get them into the columnar cell. The minute they get inside, they reform the triglyceride. So now it definitely is gunky, lardy, like the typical characteristics of a fat. So we've got to lubricate it again, because it is inside some teeny tiny little tubules throughout the columnar cell. In Anatomy 1, you called that endoplasmic reticulum. The abbreviation for that is ER. So that's where you are with these big, thick fats. So we lubricate it now with protein, a protein lubricant. And the protein is called, the protein-coated triglyceride is now called a chylomicron. A chylomicron is a protein-coated triglyceride. And now it's not sticky, so it'll move through the endoplasmic reticulum. And in Anatomy 1, you were told these ultramicroscopic tubular networks within a cell cytoplasm are not static. They're dynamic. They're making connections new to other organelles in the cell all the time. So the endoplasmic reticulum is going to come over and hook to the Golgi apparatus, the Golgi body, so that these fatty foods end up being put in that pancake hollow system of tubules. And if you remember how the Golgi worked, it would then push the concentrated products, in this case triglycerides, down to the end of the hollow tube and pinch off little membrane-surrounded secretory vesicles. That's how you secrete the fatty food from the columnar cell. And that fatty food is spit this way out of the columnar, it's spit that way out of the columnar cell and towards this big open valve in the end of, guess who? The lacteal. This is the big old lacteal in the center here. And the Golgi body secreted the membrane-surrounded fat, and so it's the chyle that's entering that lacteal. So to recap, not on this piece of paper, you sprayed triglyceride fat that you needed to digest with bile. That emulsified it, cut it up. Then lipase enzyme jumped all over it and broke it down into fatty acid and glycerol. Then you sprayed the fatty acid and glycerol with bile to make it water-soluble micelles. So the micelles entered your columnar cell in the ER. The minute they got into the endoplasmic reticulum, the ER, they rehooked to each other and reformed the triglyceride. So now you got to coat the triglyceride with protein. That's called a chylomicron. And the chylomicron goes from the ER, endoplasmic reticulum, into the Golgi, and the Golgi pinch it off in little secretory vesicles, and it gets soaked up in the lacteal as chyle, fatty food, chyle. Are you okay? All right. So you've got to memorize all those weird terms and who's bile-coded and who's protein-coded and what organelle are you in. But basically, the absorption's the tricky thing with lipids. The enzyme is a lipase, and it's coming either from intestinal crypts of Lieberkühn or from your pancreas as steapsin or pancreatic lipase. Yes?
Is there a reason why the chyle protein coats the triglyceride? Like, is there a reason?
Yeah, because these are, ER is a little tiny tube, and if you're like lard and you gum up the tube, you're not going to keep moving the protein out to get it secreted so you can take it up into your circulatory system. So yeah, it's to keep it moving because that's a tiny tube, and Golgi body is a tiny little organelle that you're trying to squish it down to the ends of and concentrate it. Yeah, it's lubricating it. Okay, that brings us to protein. So go back here. Don't get too far from this piece of paper because on the other side we're going to need to be referring to the protein page in a minute. So, here you are in your mouth. Nothing works on a protein in your mouth. So here you are in the stomach. And in the stomach, you had glands. What were the glands in your stomach called? Yes, I love you. Gastric pits. Those are the stomach glands. Gastric pits. And there were two kinds of cuboidal cells in a gastric pit. A parietal cell, which had pink cytoplasm, and that secretes HCl, hydrochloric acid, and a purple cytoplasm cell, which was a chief cell, and it secretes an inactive enzyme called pepsinogen. See the OGEN ending? That means it's not active. We're going to have to eat off a little amino acid or polypeptide chain off of one end to turn it into the active compound. So the question is, how do we activate pepsinogen? And the answer is the HCl from the parietal cell comes and helps the chief cell's pepsinogen secretion. The HCl eats off the amino acid chain, so the active form is called pepsin. Enzymes often end with IN, so this is obeying the rule of enzymes. And the pepsin acts on proteins. Now, it will not get it all the way down to one single amino acid. It will get it down to a chain of 3 or more amino acids. So this says greater than 2 amino acids. Those different combinations of amino acids are called proteoses and peptones. But you're at least that long. So you're still too big to get absorbed. You got to be 1 amino acid in order to get absorbed. So we're not done. We're just started on protein digestion. But pepsin did all that. This passes on down. You're now in your small intestine. In your small intestine, in the duodenum, your pancreas dumps in 3 different secretions. These are all zymogens, which means they are also inactive. So one of them is called chymotrypsinogen, and notice the OGEN ending, and it has an asterisk to remind you it came from the pancreas. And trypsinogen, so that's CHYMO, chymo, TRYPSINOGEN, chymotrypsinogen and trypsinogen. So this just has the chymo prefix out front. When we get these 2 enzymes turned on, they will then just lose the OGEN suffix. So this one will be called chymotrypsin, and this one will be called trypsin. But these enzymes act on the 3 amino acid proteoses and peptones and take them down to this size. So they're called dipeptides now because it's 2 amino acids bonding. I still haven't told you how we got these activated, but I'm telling you what they do when you get them turned on. Okay? Meanwhile, there was a third pancreatic secretion, but it didn't help you in this step, so I waited till over here to mention it. The third pancreatic secretion was called procarboxypeptidase. By putting pro in front of something, that's another way of saying it's an inactive zymogen. So we got to cut off that little amino acid chain. So the active form of that is going to end up being called carboxypeptidase. Carboxypeptidase. So that has an asterisk because it too came from your pancreas. And when it gets turned on, it will take you the rest of the way, but so will a group of protein splitting enzymes that came out of your crypts of Leverkusen. That group is called erepsin. That's not one chemical, that's a group of small intestine protein splitting enzymes. They are a variety of aminopeptidases. That's another way of saying the same thing, but erepsin. So what I'm saying to you is erepsin and carboxypeptidase do the same thing. They take your dipeptides and break them down to single amino acids. Are they ready to be absorbed? Yes, they are. So you'll use ATP to force all of those protein subunits, those amino acids, into your bloodstream. So what we want to look at on this page over here then is how you got those different zymogens turned on. Now let's remember one thing before we get started. Up here at the top of the page, remember you had from your stomach, you had pepsinogen. And it was turned on by hydrochloric acid, and when it was turned on, it became the active pepsin. And when you had active pepsin digests proteins down to this, which we called proteoses and peptones. So we knew that, right? We already knew that. That's how you got pepsinogen turned on, HCl. Once you got it, it could start on your proteins, and you got this far in your stomach. Now that food was passed on to your small intestine, where you were hit with this from the pancreas, and this from the pancreas, and this from the pancreas. This was inactive. How did you get trypsinogen turned on? This is you're in the small intestine, remember. This enters the small intestine from the pancreas. And so in the small intestine, you've got these, a villus. And this is your gland, the crypt of Leverkusen, right? Down at the bottom of the crypt of Leverkusen, there is a special cell called a Paneth cell. That Paneth cell secretes an activator of this enzyme called either enteropeptidase or enterokinase. You can call it either term you wish. But it is what cleaves off the little amino acid chain. So now inactive trypsinogen becomes the active form, and here's the name of the active form. I circled it, trypsin. That's your active form. But you can thank the Paneth cell's secretion, enterokinase, for that. When you get trypsin activated, it turns around and activates the chymotrypsinogen by eating off an amino acid chain on the end. And so that is what's doing the work. So you end up with the active enzyme chymotrypsin. So I circled that one. And number 1 and 2, in other words, trypsin and chymotrypsin, then act on your proteoses and peptones and turn them into dipeptides. Turn them into dipeptides. So we got one more step. We got to get the dipeptide broken down. But the other pancreatic protein splitting enzyme is also a zymogen. We've got to get something to eat this amino acid chain off the front. That's trypsin again. So trypsin helped activate 2 out of the 3 enzymes that came from the pancreas to digest protein. So trypsin turns that into the active form, which is carboxypeptidase. And now carboxypeptidase from your pancreas and erepsin, which is from small intestine glands, both do the same thing. They act on the dipeptide and turn them into single amino acids. So now you can absorb them. Yes? The active carboxypeptidase from your pancreas and the mixture of protein splitting enzymes from your small intestine both do the same thing. They take this dipeptide, split it apart, and now you're finally down to single amino acids and you can absorb them. So there are numerous protein splitting enzymes. You need to know the one from the stomach, pepsin, the 3 from your pancreas, trypsin, chymotrypsin, and carboxypeptidase, and the one from your intestinal cryptoglycan, erepsin mixture. So far, so good? Sure.
All those were called protein.
All of these collectively would be called proteases. They're protein splitting enzymes. So that's a very umbrella, generalized word. But your proteases would be pepsin, that's from your stomach, trypsin, chymotrypsin, carboxypeptidase, those 3 are from your pancreas. And then you had erepsin, which is a mixture of protein splitting enzymes, and those are from the cryptoglycan of your small intestine. Okay? Pepsin from your stomach, trypsin, chymotrypsin, carboxypeptidases from your pancreas, and from the cryptoglycan of your small intestine, erepsin is the mixture. So in addition to just knowing the name of the enzyme, you need to know what food did it start with, undigested protein, what food did it leave you with, proteoses and peptones. Get the idea? Okay. And where did it come from? Who said? Go ahead.
Did you say where it comes from, you mean like starting from the stomach?
Yeah, but gastric pit glands of the stomach, crypts of Lieberkühn glands of the small intestine, pancreas, yes, that's what I mean. Good questions. You okay on that, everybody? Okay, so when we come back next time, we still have to comment on genetic material. And that is grossly oversimplified. I'm just going to say the enzymes are nucleases, and they come from several spots, small intestine and pancreas. We're not going to go in much more detail than that right now, because you will pick up on that in other courses later, such as microbiology. So we're just going to leave them by their umbrella name, nucleases. So we're not doing a lot of detail there. So bring these sheets back with you next time. Regardless of when I finish this material, and I still have to talk to you about nerve tissue of the digestive tract and about